it's an honor to be invited to give a talk as part of this History of Ideas forum at NIAS. I'm not very sure how much what I'm going to say fits into such a topic at all, but we'll see how it goes. So very briefly, I got acquainted with the name of Turing when I was a research student, and I happened to be a student of physics, but the person with whom I worked, a solid state physicist by the name of Morrell Cohen, was getting interested in well, generally theoretical biology, and as part of that interest, he and some other people who were around there at the same time, and I'll tell you a little bit about their background in a moment, managed to get hold of quite a remarkable set of visiting lecturers who came during the time that I was a student. This was at the University of Chicago, and these lecturers were essentially all biologists. So even while a student of physics, I had a chance to listen to them and on some occasions to talk to them. One of them was a man called Louis Wolpert, and it's due to him that I first heard of Turing and heard of some of these ideas that I'm going to present here. There were many other speakers who I can even now recall rather vividly. There was Michael Gaze, who spoke about well, how certain connections are formed in the brain. Many other names. Waddington had come, if I remember right, on occasion. There were people who were also not biologists but were on the fringe, so to speak, like Prigogine and so on. So it was all in all a very exciting time where you could sort of sniff and smell something interesting and not commit yourself to doing something in it. This school was an interesting one too. The place where these people came, it had been set up by a man about whom I'll say a few words later, a person called Rashevsky, and this was for many years the only school in the entire world dealing with what Rashevsky termed mathematical biology. He later changed the name to mathematical biophysics, and it was pretty much a one-man operation. There was Rashevsky alone to begin with, then his students and postdocs and so forth, and students of students and so on. I believe the enterprise is wound up now. In fact, there's some amount of a story behind that as well. But the main reason for its getting wound up was that the way people started looking at some of the problems that interested him changed. In any case, since my degree in physics, as I said, I have been working in biology, in developmental biology, and I deal with a system called the slime mold, and this is a minute amoeboid organism which builds some kind of a society or some kind of a colony within which you can see pattern. So I do have, one might say, a professional interest in pattern formation. But the bulk of the work that I have been doing and people in the lab have been doing has been experimental. So I myself, I don't think I've done anything which would fall under the head of a Turing instability, which is what I'm going to talk about here. So my talk now really will be still as an outsider looking at something that happened and continues to happen. So perhaps I could begin. I'll start out, maybe I should keep this in my hand. I'll start out by illustrating, two kinds of things. It's so bright, but I can't. Right. This is a set of fairly common beetles called ladybird beetles. And the thing about them, of course, is that first of all, they are brilliantly patterned on their backs. It's something called a wing-like appendage that they have on the back. And the point to notice about these patterns is that they're extraordinarily varied. And as far as we can make out, this variation is not directly gene-based. I come back to what this means towards the end of my talk. Nonetheless, there is an enormous amount of variety there, and it is quite striking too. on the other hand, there are other sorts of things that one sees in nature, and I'll just flash these things rapidly on the screen. I'm sorry, that's a rather poor imprint of a zebra. You see that doesn't work. Maybe we do this. This is an artist's sketch, a platypus. I think that's a rhinoceros. Again, these things are downloaded from the web. The quality isn't terribly good. The point I want to make, which is an obvious point really, is that these things look different. Now, there's one sense in which these things that I showed you here look different, and there's quite another sense in which those arrays of beetles that I showed you look different. And that, it turns out, is one of the themes underlying what Turing was trying to talk about. So the sort of differences and the sorts of patterns that Turing was concerned with, which I will elaborate a bit in this talk, will be the sorts of patterns that you saw on the backs of those beetles. These patterns, on the other hand, which today we don't even call patterns, so what I'm saying here is the difference between a rhinoceros and a horse and a goat and a zebra and what was it, a platypus and so on. Biologists today use the word form, even though it's a kind of pattern difference, to distinguish it from the other sort of thing, which they now term pattern. And the problems of form are by no means solved today, but the general feeling is that form comes about by means of things getting bigger and bigger, by means of growth, things moving relative to each other, so there's some pushing and pulling and so forth. So that if you like to imagine life or embryonic life begins in the form of a small ball of cells, and you just go and pull that ball in some place, push it in some other place, poke something somewhere, mold it on the surface, basically play with it like almost as if it were a piece of plasticine. And then you can get a zebra or a horse or a fly or what have you. This is the general feeling in biology now, and it probably is correct. Of course, there's a great deal that goes behind this feeling about what the forces are which might mold this. On the other hand, when it comes to the kind of thing that we saw first, these patterns on the back of the beetle, it turns out that there are two rather different ways of looking at this. One of these two ways is a way due to Turing, and it's a way which is born essentially of physics and chemistry. The second way, which ultimately can be traced back, as everything else in biology, to evolution, is based primarily on genes and how they act and where they act and what other genes act at the same time that they act and so on. So one of my aims in the course of this presentation will be to try and sketch in a very cursory form that these are really rather two different ways of looking at pattern in the living world, and they offer different sorts of explanations. Well, that's just the title of my talk, and the chemical basis of morphogenesis is in quotes, because I'm not going to, this is not a talk on the paper itself, but about the paper, if you wish, meaning about its background, its antecedents, and where we have come to now. So there won't be any technical detail there. There's Arnold Turing. He was a remarkable man, and it's rather interesting for people in the computer sciences, people in the engineering sciences in general. He is thought of as one of the giant figures of the 20th century, one of the seminal thinkers of the 20th century, it is said. Personal experience convinces me that he is almost unknown to biologists. This may reflect partly general lack of knowledge of people who have made major contributions in other areas but not in biology. But in part, it also reflects a tension which has existed in biology and continues to exist in biology between people who look at biology from a theoretical or mathematical point of view and people who look at it from an experimental point of view. This too is a theme that I will touch on. Turing lived a very short life. He was born in 1912, died in 1954. His major contributions, or at least the contributions for which he is known to the vast majority of scientists, consist of what he had to say about theories of computation, about formal logic. So, for example, the phrase Turing test is associated with his name. In a very rough sense, a test which he proposed to try and distinguish between machine intelligence, if there could be such a thing, and human intelligence. Then, of course, there is the famous Turing machine, about which I think we've heard some things in this very room, or at least in NIAS, which is a formal algorithmic device to decide on whether certain propositions can be proven or not. And there is a third phrase which is associated with the name of Turing, and that's Turing instability. And this has to do with this single published contribution that he made in biology by way of a paper that he published just 2 years before his death. So the paper was published in 1952. I'll remind you that was 1 year before the announcement of the double helix, 1953. And this year we are celebrating the 50th anniversary of the double helix, and I think this is a worldwide celebration that's going on. Last year was similarly the 50th anniversary of Turing's paper, but among the biological community, it was either ignored or completely unmarked, even though people paid tribute to the name of Turing and so forth. Right. So what is this paper about? His main motive in writing this paper, by the way, so what was he trying to do in this paper? It's called, as you saw, the chemical basis of morphogenesis. Morphogenesis is an old term, basically meaning the genesis of form, of morph. As I told you, today one wouldn't say that what Turing dealt with was the genesis of form so much as the genesis of pattern. Nonetheless, we'll stick to this. So he was trying to explain the origin of pattern. His aim in doing so, this doesn't come in the paper itself, but it's known on the basis of published records and personal reminiscences. His aim in doing so was, if you wish, materialistic, meaning he wanted to, as he puts it in writing to a friend of his, he wanted to refute, he wanted to disprove the so-called argument from design once and for all. Now this argument from design, as it suggests, is that the living world was designed, as simple as that, and designed by a creator, designed by a god, what have you. And the phrase itself, the phrase argument from design itself, comes from a rather famous book by a gentleman by the name of William Paley, and I've copied what Paley said there. I'll read through it. It's a bit long, but nonetheless it's worth reading. It's very good prose. Paley says, in crossing a heath, suppose I pitched my foot against a stone and were asked how the stone came to be there, I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps be very easy to show the absurdity of this answer. So you could say it's been there all the time, and you would probably be right. But suppose I'd found a watch upon the ground, and it should be inquired how the watch happened to be in that place, I should hardly think of the answer which I had before given, that for anything I knew the watch might have always been there. Yet why should not this answer serve for the watch as well as for the stone? Why is it not as admissible in the second case as in the first? And now he comes to the answer, why not? For this reason and for no other, namely that when we come to inspect the watch, we perceive what we could not discover in the stone, that its several parts are framed and put together for a purpose, e.g. that they are so formed and adjusted as to produce motion, and that motion so regulated as to point out the hour of the day. That if the different parts had been differently shaped from what they are, of a different size from what they are, or placed after any other manner or in any other order than that in which they are placed, either no motion at all would have been carried on in the machine, or none which would have answered the use that is now served by it. The inference, something else in between which I have removed, the inference is inevitable that the watch must have had a maker, that there must have existed at some time and at some place or other an artificer or artificers who formed it for the purpose which we find it actually to answer, who comprehended its construction and designed its use. So, very, very rational argument, makes sense and so forth. Now Paley goes on to say, look, if this argument holds so much weight in the case of a mere watch, how much more true must it be in the case of a living creature? Because it would be absurd to say that it holds good for a thing as complex as a watch, and say that it doesn't hold good for something vastly more complicated like a living creature. Now this is an argument which really influenced a great many people, And if you read Darwin's life, you see that Darwin too was convinced by this. He had to read Paley as part of his theology in Cambridge, and he came back convinced. Of course, he changed his mind later, but that's a different story. So Turing's aim, as I said, was very materialistic, and he said, I want to show that this can't be true. But he said, I want to show this from the principles of physics and chemistry. And the reason Turing did this, and I'm going to come to that later, is because he explicitly did not like evolution, by which I mean did not like evolution by natural selection. Now, among evolutionary theorists of the post-Darwinian period, there were two groups or two classes of people. Those who saw that natural selection quite obviously was the answer to the complexities of the living world, and those who said that maybe there was something else living behind the sort of order that you found in living organisms, especially something behind the sorts of shapes that you found in living organisms, which probably had a more basic explanation, one based on, as I said, physics and chemistry. And this was a long kind of tension which persisted for very many years. And in fact, it's rather curious to see that it has come up again in evolutionary biology, with people claiming that you may have quite valid explanations for biological form which do not depend on evolution, do not depend on natural selection. I come to that later. So this, so much for Turing's aim. Now to go back to that previous slide, what did he do in this paper? So his aim was to refute the argument from design once and for all. In the paper, he does something really very simple and straightforward. He says, ultimately, if you look at a cell, or if you look at a body, if you look at an organism, what it is a bag of chemicals. And he says it makes sense to think of whether, given a bag of chemicals, and given that chemicals do certain things that we are familiar with, there might be any way at all that within this bag you might get spontaneously arising, right, no external agent, spontaneously arising, the kind of shape or the kind of pattern that you are finally interested in. This was the question that he tried to ask. And while doing this, he went through a number of steps. So he said, to begin with, chemicals react with each other. So you have to write down some equations of chemical reaction. And he used the standard equations of chemistry based on the laws of mass action. Then he said, there's something very important in chemistry called catalysis. So probably there are chemical catalysts within the cell. And catalysts, of course, influence rates of reaction and give you effective rates which don't look like simple mass action terms. And then he borrows these laws of catalysis, which we today call laws of enzyme action, from Michaelis and Menton, simple kinetics. And then he says, this is rather prescient if you think that this was written 1 year before the double helix. He says, we do know that there are things called genes, and there are good reasons for thinking that genes probably influence form, but they probably influence form at so many steps removed from the form that you actually see, that the ultimate cause, if you wish, of what you are seeing consists of this bag of chemicals and the way these chemicals talk to each other. So he talks of genes which catalyze the production of certain chemicals, and he coined a term here, he called them morphogens. This is a term that we use even now. So in a rough sense, form causing chemicals, morphogens. So there are genes which catalyze morphogens, and he says these morphogens probably are also catalysts, which is an interesting concept. They catalyze other morphogens and so on down the chain, until he says you finally have to come to small molecules which can move from cell to cell or move within the tissue and then engage in these chemical games, if you wish, and come up with something. Right. I will skip over points 4 and 5, they aren't very important now. What is interesting about this paper, by the way, the end of this paper is that he shows you how patterns can arise, and I'll show you some specific examples later. What is interesting about this paper is that it pretty much stands on its own completely. In fact, he could have done away with references altogether. There are only 6 references in this whole paper. 3 of them are technical. They are not terribly interesting. One is to James Jeans. He uses a formula for spherical harmonics, so he says Jeans, electricity and magnetism. One is to, a pair of people called Davson and Danieli to get a number for permeability. So that too is a number that he picks up. And then 3 are to predecessors of his who had interesting roles to play in this story. C.M. Child, who is a man not known to many biologists now, but who first developed the idea of what is called a gradient in developmental biology today, and said gradients must underlie form and pattern. Then someone called Darcy Thompson, on whom I'll spend a little bit of time. And finally Waddington, who he cites for his use of the term evocator. And Waddington had some interesting things to say about Turing after the paper was published, to which I come as well. So what I want to say here is that the paper itself consists of asking, what is the simplest form of system in which I can put bags of chemicals to work? I allow them to react with each other, and of course chemicals can move from place to place. If their concentrations differ, they can diffuse. So I put in chemical reaction, I put in diffusion. Don't specify any particular form for these things. Given these conditions, what are the requirements that I need to put on the chemical reactions to get interesting forms? And of course you have to define what you mean by interesting forms. Turing looked at something that today is modeled by zebra stripes, but he took an equivalent one-dimensional system, a ring of cells. So he said from a ring, the equivalent of stripes would be lobes, right? And he said, look, this looks like the tentacles of a hydra. So that's one specific kind of pattern that he came out with. And he also showed that you could get what are called today coat color patterns on mammalian coats, which are really very common. If you look at a dog or a cow or a cat around, you see the body pattern. So these kinds of patterns he was able to simulate quite easily. And then he went on to show that you could get other kinds of patterns too, but didn't model them in detail. So he ends his paper basically by saying, I set out to do something, and here it is, I've done it. So this is possible. And he says, given that this is possible, it would be surprising if nature had not made use of it, because I've not made any special assumptions. This is just chemistry. In fact, there's very little physics per se in this paper.
We have to give some very general property of what is the equation we use here. Obviously, you're not going to put in some any specific values for chemical reaction rates and so on. So is this something very powerful mathematics, or is it?
No. The mathematics is, all depends on what you mean by powerful, right? It's
obviously able to make some general deductions from a set of equations in which the parameters could be some
quite right. But the equations or the actors in these equations have to satisfy certain conditions, which I will come to.
And then it's powerful.
Okay. Now I want to spend some time. So the point I wanted to make so far was that Turing did this, carried out this demonstration, and the way you read the paper today, you get the feeling this came out of nowhere, and this may very well be true. It may very well be the case that Turing really wasn't influenced by anyone. By the way, I forgot to mention this. Excuse me, I had a list of names of people who, historically speaking, even though they did not quite anticipate Turing's thinking in detail, should be counted among his predecessors. I seem to have left that slide, but I just mention it for the sake of completeness. Not too many people. So I'm speaking of people who said that in some sense, to use the same phrase, you could think of organisms or cells as bags of chemicals, and this bag of chemicals could give rise to differences in space, which could foretell the sorts of differences that really interested you later, stripes or spots or these, speckles on the back of the beetle and so on. Amazingly enough, and I didn't realize this until I had started preparing for this, the first version of this talk, T.H. Morgan seems to have thought of this a long time ago. So the earliest citation I could get was to a paper by him dated 1915, where he talks of not really pattern in this sense, but about polarity, about differences between one end of an embryo and the other, if you wish, and says that this kind of polarity, which is really a qualitative difference, ultimately you get a head at one end and a tail at another, could come about because of a quantitative difference in chemicals, which could spread spontaneously. What he was talking about was something called the flatworm, on which he also carried out a great many experiments, regeneration experiments, and he tries to explain these in detail on the basis of what we today would call a spontaneous gradient of chemical concentration, some kind of evocator or morphogen concentration from head to foot. So he...
Morgan and centimorgan.
Same Morgan and centimorgan, quite right, yes. So that was one predecessor, if you will. The other one was a name that I mentioned briefly earlier, which was Rashevsky. Rashevsky was this man who set up the School of Mathematical Biology in Chicago, and there is available even today, I think it's out of print, but is still available, a 2-volume collection of papers by him. He put them together and made it into a book called Mathematical Biophysics, and you will see everything on earth in these 2 books. In fact, there's the he calls everything a mathematical biology of something or the other. There's the mathematical biology of cell division, there's the mathematical biology of movement, mathematical biology of regeneration, and believe it or not, he even has a paper on the mathematical biology of automobile driving. So you can see that Rashevsky, went to, there was some kind of excess in his zeal to propagate mathematical biology. And he also was a bit of a stubborn man, refused to see himself as being anything other than the only correct thinker of many of these problems, rubbed many people the wrong way, and for a whole variety of reasons, many of them having to do with the personality of Rashevsky himself, this enterprise stuttered along and, as I said, died fairly soon after Rashevsky himself left the university, didn't get going. I should mention a book at this stage which perhaps the NIAS library should get hold of. It's by a lady called Evelyn Keller, came out just a year ago. It's called Making Sense of Life, and she has a very fine analysis of 2 things really. One, this tension between people who come from the outside and tell biologists what biology is all about, and these are primarily mathematicians and physicists, and why biologists don't feel too kindly towards such advice. And the second thing is precisely why this enterprise of Rashevsky's was such a disaster. It's an interesting book and tells quite a bit about how science actually works. So those are 2 names. The third person, and on this man I'll spend a little bit of time, was Darcy Thompson. He was a remarkable person. He too has a book which has been considered a masterpiece of prose. It's called On Growth and Form, and it's there in the Institute library. It probably is in this library as well. And Darcy Thompson now, in contrast to Turing, if you wish, who came much later, was out and out an anti-Darwinist. And for him there was something intrinsically wrong in the Darwinian notion of natural selection giving rise to tiny modifications which affected perhaps small parts of a system, and then in the course of time finally gave rise to a different form or a different morph or whatever. He seems to have had what we would today call a Gestalt point of view. He said it's meaningless to think in terms of parts of organisms or parts of bodies, and what really counts is the whole. So you really need theories if you want theoretical explanations of biological form, theories which explain the whole. And he said you don't need to go very far to search for such theories because physics and chemistry offers them to you. Here's a picture of Darcy Thompson. That's a parrot on his right hand, and stories about him say that he was a fixture at the University of Aberdeen and in the town of Aberdeen, because you could always see him with his parrot, and it may well have been the same parrot throughout. He lived to be 88 years old and created one of these world records which will never again, I think it's fair to say, be surpassed. He held a professorial chair for 64 years at the University of Aberdeen, and held a chair that long. Okay. Right. Now I'll spend a couple of minutes on what Thomson's point of view was. To begin with, as I said, it was that it's to physics that you must look for the solution of problems of biological form. Because he said there's no special reason to think of material entities, living bodies if you wish, living matter, as being any different in its physical properties from non-living matter. And he said that being the case, why don't you try and see whether the sorts of explanations that are there that might give rise to shapes in non-living matter. And in a general sense, these would be explanations based on things like minimizing energy, setting up equilibrium structures and so forth. Why don't you see if that kind of explanation works for living matter as well? The second part I've made already. He was strongly anti-Darwinian, and in fact this comes through time and time again if you read on growth and form. Of course, the point he also made was, he said, look, no one would question the possibility that form as it develops between fertilization and adulthood, or between fertilization and birth, embryonic form, must have to do with forces, with pushing, with pulling, with chemistry and so forth, right? What you would call a conventional physical cause-based causal explanation. That being the case, why shouldn't you assume that a similar explanation might also hold good for the major changes that have taken place in evolution, not over one generation, but over hundreds or thousands of evolution. He didn't like genetics one bit, and here too, there's an interesting, that's a different story altogether, but there's an interesting parallel with the whole school of thinking in the post-Darwinian, of course, but post-Darwinian and post-Mendelian days, which said that genetics is all and very good and is fine in its place, but we are interested in evolution, which is a different thing. And the genetics that Mendel talked about, or the genes that Mendel and his rediscoverers talked about, have nothing to do with evolution, which is what we are interested in. Historically, this has forerunners going all the way back to German idealists, people like Goethe and so on, who said that form must be studied as shape, and form must be studied as something in its own right, with its own laws, with its own inner justification for existence, this sort of thing. And Darcy Thompson, he was not an idealist of the Goethe type at all, by no means. He was out and out a materialist. But nonetheless, the line of thinking that he adopted was similar. I'll just flash here examples of the kinds of things that went into illustrating, that Thompson used to illustrate his ideas. So these are skull morphologies of a chimp, a human, and a baboon. And what he's trying to show here is that simple geometrical transformations, geometrical mappings, can convert one to the other. So he said, this is mathematically possible. I can do it here and I can show you here. So this is the way these things must have happened. So there must be some underlying laws which have made this happen. So in a very crude sense, you can think of these as three possible equilibrium structures of the same sort of whatever underlying substrate, bony plates in this case. And either of these equilibria being possible depending on maybe different starting conditions or different constraints outside or whatever. So there's no need to posit evolutionary change or natural selection. Of course, one won't speak of natural selection as leading you from a chimp to a human at all, in any case, but you can think of that chimp skull as being somewhat similar to the skull of a common ancestor of the human and the chimp, and the argument still holds. Beg your pardon? No. And this is another example, two completely different genera of fish. By the way, those of you who are interested in playing on the computer, you can actually do this yourself now. So there's a Darcy Thompson website, like a website for everything else, and you can go and choose rates in which things move in this direction, that direction, and it's rather interesting. You can get pretty much anything from anything else within certain limits. Right, now I come back to Turing, and I'll take 5 minutes to try and tell you exactly what he did, just so that you get an idea. Okay. One of the things I didn't mention but should have is the following. Among the predecessors of this Turing paper, completely unknown to Turing evidently, were a bunch of famous Russian physicists and mathematicians, Kolmogorov, Piskunov, and Petrovsky. And what they did, I've been unable to understand for what reasons they went into this, was to work with a system consisting of one component, so you can think of it as a chemical. In fact, they suggest various analogies. One component which diffuses, right, and which also is subject to some form of production, a single component. And they managed to show that this particular equation, which is exactly like Turing's diffusion reaction equation, except that there's only one chemical doing the diffusing, this thing, given certain conditions, of course, on that function that you have and so forth, and you need to work in theoretically infinite space, this kind of thing can give you traveling wave solutions. In other words, it can give you, if you wish, a packet of activity which keeps moving from left to right constantly. And I mention this now because this equation and this way of looking at traveling waves has gained a resurgence has come into its own recently in ecology of all areas, where the waves can be traveling waves, for example, of seeds. So people who are interested in looking at historical patterns of forest successions are applying Kolmogorov's idea. It's interesting to mention now that Turing's theory needs a minimum of 2 chemicals. So this is a qualitative result that he came up with. It's rather easy to show for us today that the sorts of patterns he was looking at don't work with one component, with one chemical, need 2, and of course 3 or 4 or so on would be better. And these 2 chemicals very qualitatively again play the role respectively of an activatory chemical and an inhibitory chemical. And what does an activatory chemical mean? An activatory chemical is something which catalyzes its own production and also catalyzes the production of the second chemical. An inhibitory chemical is one which inhibits its own production and inhibits the production of the activatory chemical. So this is basically it. In today's language, you would say that these showed positive and negative feedbacks. And the second kind of condition you need, qualitative condition you need, is that these 2 chemicals have to move, or technically have to diffuse at different rates. So the activatory chemical has to diffuse much slower than the inhibitory chemical. So if you have a local center of activation and inhibition in one place, the inhibition spreads much faster than the activation. The negative influence spreads much faster than the positive influence. So this is it really. And the entire bag of tricks that comes out as an output is based really on only these qualitative considerations. Okay, how does it work? Extremely simple, and you can I can describe it verbally. So in these 4 sketches, which you must read from A to D, the continuous line shows the spread in concentrations of the activatory chemical. The dotted line shows the spread in concentrations of the inhibitory chemical. The central assumption in Turing's way of looking at these things was to say that in the beginning, you have no difference from place to place. So you have no special place from which something starts. But purely by accident, what you would call fluctuations, there's some place where some activity begins. And this is something which anyone who looks at chemistry will agree is plausible. So there's a temporary fluctuation, and Turing says, let us look at a fluctuation which gives you a small hillock kind of increase in the activatory chemical. So that's in A, spontaneously occurring. Of course, in ordinary chemistry, day-to-day chemistry, the kinds of things we do in the lab, this happens all the time, but then the fluctuations go down and so forth. However, given the special properties of these 2 chemicals, that small hillock keeps growing, which you see in B. In addition to growing, it creates a second hillock. This is a hillock of inhibitory chemical concentrations, that's the dotted line, right? Because of the property of activating itself and the inhibitor. Now notice that the inhibitor spreads much faster than the activator. So as you move away in space from this local place of activation, you find that inhibitor levels are higher than activator levels, right? And because they are higher than activator levels, they ultimately succeed in pushing both themselves and the activator down, which you see in D, which means now that if you move away from this peak that you see, please look at curve D, if you move away from this peak, even if further fluctuations occur there, they won't be sufficient to raise the activator above threshold. It's been depressed so far. So it's as simple as that. And all that this shows, of course, is that you get one localized peak of activity and one localized peak of inhibitor. If you move sufficiently far away from this peak, sufficiently far meaning far enough for the inhibitor itself to have decayed down, then of course a second peak can arise and a third peak and so on. In short, what you can get from this kind of a model pretty much automatically is a series of repeating peaks of this kind, spaced evenly, which is essentially like the stripes on a zebra's back. Of course, the other things that you can get need qualitatively similar arguments, but in detail slightly different. So this is what Turing showed. Right. Now, are there examples of Turing patterns? The kinds of examples that are advanced are this. This is a copy from this famous painting by Mansour. So the stripes on the zebra's back. Something that's found in a creature that I work with called the slime mold, which I mentioned earlier. And what I want you to notice here is that there is this ball of cells going from A to B to C, where spontaneously you find a certain number of cells which are evenly spaced around the perimeter, acquiring special properties. So the black color there simply means that you can do something to do something to these cells so that these things show a particular color and the other ones don't. And you can look at this in rather detail. So the A, B and C are hand-drawn sketches. The A, B, C, D below are actual photographs. And this particular ball of cells, in fact, gives rise to protrusions, in this case 5, which sort of look like a starfish coming up. Okay, so this is a nice example of what might be a Turing pattern. So spontaneous generation of this kind of patterning, and that's in a ring, as I said, except that this is a 2-dimensional ring. Okay, now I come to something remarkable which has been shown very recently by a person called Shigeru Kondo in Japan. He has now, there's a bit of a story in this which is rather interesting. Kondo got interested in Turing patterns. He's a developmental biologist and he works in a place called the Riken Institute in Kobe. He got interested in Turing patterns. To preempt myself a bit, I should tell you that to date, no one has given a cast iron example of a pattern in biology which has to be a Turing pattern. In other words, no one has said this is chemical A, this is chemical B, these are the reactions, and with paper and pencil or on my computer I can show that these reactions give rise to these outcomes, and here is the outcome shown in real life. That hasn't been possible yet. Keep that in mind. So this is where Kondo's thing becomes interesting. What he has now, what he did was to write to a whole lot of mouse labs around the world, people who breed mice for laboratory practice, and asked them whether in their litters they ever came across a line which showed funny colors on the body. Okay, not the normal mousy color, but strange colors of this one kind or the other. And apparently from a place called the Jackson Labs in the US, which is a fairly large stock center as well for mice, he got a reply once saying, yes, we have one line where this thing doesn't seem to be doing very well because it shows one color one day and some other color some other day, and
different litters on different rays, or same mouse?
Same mouse. So the litter showed this property. A single mouse seemed to show color changes in color.
A single mouse in a given litter. Change in time.
As a function of time. So that, generally when these kinds of things happen in the lab, you say, that beast is sick and put it away and then get back to your usual kind of thing. So Kondo immediately wrote to them, got the mouse and this and that, and did some very clever breeding experiments, conventional genetic kinds of experiments, established that this was due to some kind of a genetic change, due to a mutation and this and that. So he has a number of mouse lines now with him maintained. The mice, by the way, seem completely normal, healthy and so on. They're not sick at all. But they show interesting body patterns which are reminiscent of the kinds of things that these reaction diffusion mechanisms exhibit, or Turing instabilities exhibit. By the way, the word Turing instability is because these fluctuations arise spontaneously and give rise to pattern. Okay, so this is one of those mice which, as you see, going from left to right over a period of 30 days goes from a black coat to some kind of slightly mottled looking coat to an almost completely white coat, right, and completely black again. And this cycles. Mice don't live all that long, so you don't get too many cycles, but he says he's got 2 cycles in one particular case. Right? By the way, we know what the colors are due to. They are due to deposition of melanin pigment. So there's a cycling effect, there's an oscillation in melanin pigment deposition. I didn't mention this, but this is the time to do so. One of the outcomes that you can get from a Turing model, if you fool around with parameters sufficiently, is this clock-like behavior too. You can actually get oscillations without spatial patterns. The period, he says, is roughly this. Yes, roughly 1 month. Yes. Which one? Just one moment. As far as Turing's prediction goes, the final pattern is a stable pattern. It's an equilibrium pattern, right? And that is referring to the zebra stripe kind of thing, or the, what did I say, leopard spots if you wish, or giraffe spots, or what have you, whole lots of things. Okay. In addition, so these are the things Turing worked with explicitly. In addition, he pointed out that by playing with parameters, you could get other kinds of things. You could get homogeneous patterns where things just went up and down, right? Entirely temporal oscillations with no spatial pattern at all. He also showed that you could get traveling waves, right? You need to play with things here. For traveling waves, it turns out, for example, that you have to fine tune things if you have only 2 chemicals, but you put in a third chemical and it works rather easily. You have to do some of these things. Okay. Well, he was very well aware of electrical instabilities. In fact, the explicit analogy he draws to his Turing instability is what happens in an electrical oscillator. You know, and he spends a couple of sentences to say, look, any of us who is familiar with noise in an electrical circuit with an oscillation finds it difficult to understand how it starts. It's easy to understand how it keeps going once started. And he says, this is exactly the problem that I have here right now. I want to explain how chemical instabilities start. And then.
But actually, let me need at least 2 degrees of freedom, even for electrical oscillation. So you would need 2 chemicals, right?
With one, the thing doesn't work. That's fairly clear. It's very easy to show. Yeah.
I was just going to say that there's a point about hydrodynamic stability, which are spatial.
Yes.
And of course, Turing was in Cambridge.
Yes.
And some of this classical work on how patterns form
Yes.
was all done in Cambridge, for example, by J. Taylor. And there, basically, you get spatial patterns by a balance between diffusion and inertia. And so, from one point of view, he's getting instabilities from the kind of equations that he had. It's not a mathematical surprise.
Right.
It's not a mathematical.
No.
And this question about patterns stabilizing, that once again, will happen if there is non-linearity in it. I mean,
Yes.
It begins as a linear instability,
Yes.
but it will saturate
Yes.
if there's the right
Yes.
kind of nonlinearity.
Yes.
Turing systems were non-linear.
Yes.
So, yeah. But there are these cases where you can get different kinds of behavior.
Yes.
The systems are non-linear.
Yes. Right. So this sketch, again from one of Kondo's lines, is a mouse which shows traveling waves along its coat. So he has a whole menagerie of these things. There are some, there's a zebra mouse, for example, which shows things. This is the traveling wave mouse. He has patented these, of course. These days you have to patent everything you have. So, and there is the other mouse that I showed you a while earlier, which gives you these purely text. Yes.
See, living systems are in some kind of a steady state.
Yes. Yes.
So, the equilibrium pattern doesn't receive any kind of energy input. Now, however, a steady state pattern which can also be stationary is constantly receiving some input. The question I'm trying to ask is that what Turing does, is looking at a chemical reaction or something like that.
Let me put.
The reaction has to be endothermic or exothermic. It was going to come in and out of it. What do you say about that?
I won't answer that question because I don't know the answer, but what I'll tell you is that the systems he works with are open systems. They're not closed systems, certainly, otherwise the thing won't work. Right, so this is his model for these mice, you see, different kinds of things. And it's amusing, what he has there in the background, some of you in the audience will recognize this. So he has superimposed a cartoon of this mouse on what's known as the Zhabotinsky oscillation or the Belousov-Zhabotinsky oscillation. And it's rather strategically positioned because his detailed modeling tells him, if you look at it carefully, that the origin of these traveling waves, so what he calls the pacemaker, is in the armpit of the 2 forearm pits of this mouse, and that's where the waves travel out from and so on. So I just show that for fun. So the waves that you see at the back have nothing to do with the mouse, but it's to show that you can use probably similar arguments to describe these two. Okay, now I come to Turing today, and for some reason, my watch may have stopped. Do I have time?
Yeah, sure.
Oh, there is something there which good. So 5 or 10 minutes more. Right. I come to the status of Turing today. Now this is rather curious, as I said at the beginning. Among theoretical biologists, he's an icon. He's the closest, I think, that they have to a saint. And it's come to such a pass that it's almost impossible to write a paper dealing with any aspect of pattern in biology today, theoretical paper or modeling paper, dealing with any aspect of pattern in biology today without giving a citation to Turing. And you can see easily if you look at these things carefully, that half the time it's entirely unwarranted. So he's invoked more than made use of. And then of course there's a great industry in this reaction diffusion equations game, and there of course Turing is quite rightly the first person who comes to mind. Now, so this is again from the web. You get an idea of how the number of people who make use of Turing's name in print keeps going, and whoever it is who did this also tells you that this is an exponential growth with a doubling time, he says, of 6 years. So you can make what you want of it. What happened in 75? I need to think of that a bit. Yeah. Okay, now let's come to how we should see these things today. Among biologists, as I said, there was hardly any response to begin with to Turing's paper. Almost none. It certainly didn't create more than a flutter. There's one prominent exception to this, however, and this was this person, Waddington, C.H. Waddington, who was a developmental biologist with a deep interest in theory, a geneticist and developmental biologist with a deep interest in theory. And Waddington wrote a book not long after Turing's paper appeared, 10 years, but in this sense not very long after. He wrote a book where he devoted a great deal of time to Turing models, to Turing's idea, and that's the first time that a serious biologist actually tried to see what Turing had done and tried to ask whether it was meaningful. And the conclusions Waddington came up with were rather negative for interesting reasons. So this book was published in 1962. I'll just read it aloud. Turing seems to have a somewhat scanty knowledge of biology, and like many of those unfamiliar with the facts of embryology, he thought that its problems, which are difficult enough in all conscience, were even harder than they actually are. And then he goes on to say, we probably do not in practice often have to deal with completely homogeneous systems in which one can appeal only to stochastic processes to generate form, as Turing did. This, by the way, continues to be an objection raised by biologists, the objection being very simply the following. Turing's argument was that I don't want to make any special assumptions about the system to begin with. So mathematically speaking, I deal with something which is homogeneous and isotropic. And from this, given only chemistry and diffusion, I produce pattern. And what Waddington is saying here is that you don't need to do that because the egg itself is a very carefully packaged entity of chemicals with different chemicals being in different places quite often, and these being chemicals of the same sort, evocators if you wish, or things which lead to the appearance of form. So what Waddington is saying, and this is a fairly common response among biologists, is that nature has rigged the system in your favor. So why are you going to so much trouble to pretend that none of it exists and then say that you can pull this rabbit out of a hat? You don't need to do that. Right? Inhomogeneities are there to begin with, anisotropies are there to begin with, and in some sense, in not the obvious sense of course, in some sense the egg carries within it some amount of information about the shape of the embryo that is to form.
But we wouldn't accept the instability of
Who? Waddington did, but I come to that now. Waddington had no objections with that. His understanding of what Turing did was sophisticated enough. Okay, so this is one thing that he said. Then there are two other objections that Waddington himself raised at that time, and these are objections which have continued to be raised since then. One is that these Turing patterns do not exhibit what people call size invariance or scale invariance. It's a commonplace that biological patterns look pretty much the same irrespective of the size of the object that you're seeing. You know, a small zebra has the same sorts of stripes. I speak qualitatively here as a large zebra, and so on. This is a property that Turing patterns don't exhibit. In fact, they show a rather detailed sensitivity with respect to total size. There are ways of getting around this problem, and mathematicians and physicists have suggested ways of getting around it, but nonetheless this is a problem which one has to keep in mind. The second question, and this may relate to what Vikram was asking a while earlier, is that development itself is dynamic. So this pattern not only must be a pattern for this particular size of the embryo, but must continue to be a pattern for the embryo as it keeps growing, as it keeps getting bigger and bigger. And it's not obvious how you incorporate that into the model. In other words, not obvious how you incorporate growth into the model. I'll tell you very simply why these are problems. It's because, as I said earlier, the Turing pattern ultimately depends on a high spurt of activity in one place, no activity possible near it, and then a second spurt of activity being possible at a sufficient distance from the first place. The distance between these two peaks of activity is what's technically called a chemical wavelength. Wavelength simply because it is one period of a repeating structure. Chemical wavelength because it's something natural to the system and its dimensions depend only on the chemical rate constants and the diffusion constants and things of that kind. So the point is, if you have a chemical wavelength which is intrinsic to the system cannot regulate. It's rather easy to see because you have a natural scale in the system already. It's not a scale which accommodates itself to total size. Okay. The third kind of problem, and this I mentioned a book by Evelyn Keller a while ago. Perhaps I should. The third kind of problem, and this is something that has I think been brought up again and again and again, which Waddington did not think was a problem, but many other biologists seem to, is this business of a spontaneously arising instability. I won't spend more time on this except to say that Keller discusses this in great detail. She says that there's something intrinsically fishy. She doesn't use the word fishy, I'm saying this, but basically something intrinsically fishy to a biologist about saying that something happens, but there's no immediate cause, there's no culprit for what has happened. It's an instability which has come up spontaneously and given rise to this pattern. Now there are many other issues that come up when one tries to analyze when one tries to analyze the differences between the way Turing tried to look at patterning and biologists on the whole, we try to look at pattern on the whole today. For one thing, developmental biologists, the kinds of biologists who are interested in what happens during embryonic development, during embryogenesis, try to look for explanations where you have cause and effect chains. So you see something, and the question you ask experimentally is, how can I design the right experiment to figure out what has caused this? And then you come up with some sort of an answer, and usually today the answer turns out to be in terms of genes. You say this particular gene was active in this particular region of the embryo, and you can get more complicated than that. You can say, in conjunction with these two other genes, it did such and such a thing. That is why I see what I'm doing. And of course, one can go back and say, why is that gene active in that particular region of the embryo? You push back the causal chain a bit. This has been the operational method of developmental biology, certainly in the past 20 or 30 years. Before that, of course, there was a way of looking at development.
I must say that this has not been the operational method for biology for a very long time, because biologists are perfectly aware of these underlying patterns in which the genes are acting. They were fairly sophisticated in 30s and 40s to know this. So I don't think that it is foreign to a biologist's mind to think of patterns.
No, I didn't say that. Let me.
Yeah, you're talking of some pattern which exists, which is not created by genes, but which, let us say, a theoretical approach can tell you that it can be created.
Yes.
Since child's time, biologists have been aware of the fact that such patterns exist in which the embryonic development
True.
takes place.
True. I wouldn't.
No difficulty and no conceptual difficulty in saying that.
Okay. Let me try and continue with. Perhaps we can discuss this later with the point that I'm trying to make. If you look at a conventional developmental biology approach to patterns, now look at a standard book or look at publications which come.
I'm not talking about the standard books. I'm talking about what the thinkers have been.
Absolutely. There I
thinking.
I agree with you.
Their proof will tell you.
Okay. I agree with you 100% there. So okay, then there's no.
What we're talking about how it is explained in
Okay.
public.
Yes.
Or how
Yes.
people can lecture.
Yes. Okay. No. Then there's no dispute. I go along with that. So to come back now. So the conventional view, if you wish, the textbook kind of view, if I might repeat, of indeed much or all of biology now, and it's getting increasingly fragmented into smaller and smaller units. But the conventional view until quite recently was to talk in terms of genes. And in fact, there are good biologists who have said, if you want to understand development, you must understand why a particular gene acts in a particular place at a particular time. This is the operational framework within which you can make sense of, certainly of embryology, of developmental biology. This view, of course, is completely different from Turing's point of view, as you can see, or Darcy Thompson's point of view. And I think there are many, many reasons for this. One, of course, is this business of wanting an immediate causal explanation and wanting a chain of cause and effect relationships. The other thing, which is rather interesting, is that the whole philosophy of mathematical modeling, in physics at least, and by extension in the physical sciences and among those who try to carry out in the biological sciences as well, the whole approach is to try and, if you wish, extract the essence of a system, to try and simplify a system, abstract only as much as is necessary to make whatever point it is that you are trying to make, and ignore everything else as irrelevant. Traditionally, or at least among the vast majority and run-of-the-mill biologists, I would submit that this is not the case. Rather, the philosophy of biology has been, can I try and figure out everything that possibly plays a role, possibly influences the thing that I'm looking at? And then can I figure out how it influences it in detail to give the end result that I have? So in a very slightly misleading way possibly, but in a catchy phrase, one might say that whereas physicists traditionally try due to various different reasons, and then some of them happen to be more advantageous than others under certain circumstances, and so on and so forth. So certainly over long time scales, it's impossible to provide a cause and effect explanation of what has gone on in the natural world in the same sense as you can, let's say, in explaining the evolution of a chemical reaction or a hydrodynamic flow pattern or what have you. And I think this notion of messiness is tied up with the idea, which is also not by no means false, that these things that are evolving ultimately, these things that are changing due to natural selection ultimately are genes, DNA sequences if you wish, what are they? And the number of those genes that can influence any particular trait, be it a physical trait, a behavioral trait, a form of pattern, a biochemical trait, what have you, the number of those things that can influence this trait is enormous. That being the case, I would in some way be losing something essential about the system if I try to, brush away a great deal of what I know actually forms part of the system, and then say I will look only for sufficient causes to explain the system. I hope I'm making the.
It's an important point that you are making, and there has been an awareness of this for a long time. There's a real difference between a physicist. So I was going to ask you this question. I'd like your comment. Why is it that a paper like Turing's paper that you mentioned has been so singularly uninfluential in biology? Maybe Evelyn has picked it up now and will make some comments, which may be of historical interest, but it actually has had no influence on the course of development of biology. So is there some reason behind it? Not that people are not aware or not that they are not sophisticated, and that is very good. Why is it? So there is this point that the question behind that raised that physicists are always thinking of equilibrium systems and thinking of laws in terms of equilibrium system and thinking of minimalist explanation. Whereas biologists know that the biology doesn't work with minimalist explanation. Very often actually minimalist explanation prove to be wrong. And developmental biology, more recent years, is a very good example of that. You had all these very sophisticated theories, but the actual explanations turn out to be always messier and a combination of more. So they are intuitively aware of this, that the real thing is more than your theory is trying to encompass. And they are interested in what they. I mean, there is a point behind. So Delbruck always said that, as you say, run of the myth is he doesn't understand that you are dealing with a steady state, steady organism, steady state. I mean, that's the real problem.
Yes. No, I would go. I would just carry on. I would carry on with what you have said to say that the thing that biologists are completely convinced of, and you have to do biology to see that this must be true, is that living creatures are historical creatures. Now, physicists are very uncomfortable with saying that to try and understand some system, you have to look at its father and grandfather. This is nonsense. You look at what there is here, and this is a totally different point of view, completely different point of view. And I think the historical approach is embedded deep within evolutionary notions, and we know it is true. This you can't. So there's this dichotomy or contrast.
Well, you have said this, and you quoted many people who say this about the kind of conflict, how these two views are totally different. View that it might be due to instability and view which goes back to the genes. And there's something there in that argument which I'm missing clearly, because to me it doesn't necessarily seem so contradictory. Even if Turing is right, Turing may or may not be right, even if Turing is right, all he is describing is the machinery of how it operates. He is saying chemical A can react with chemical B, and if you have the right kind of diffusion, you can produce this kind of pattern. But he can't say, based on the theory, what chemicals there are going to be, or what concentrations there are going to be. And is it inconceivable that genetics selects for certain kinds of chemicals or eliminates others because they won't survive usual evolutionary explanations? But what Turing is describing is really the end stage there. I mean, it's like saying, well, there are of course explanations for why, for example, we're all what we are, but that doesn't mean that if I'm lifting a weight or running, I defy Newton's laws.
Yeah. No, I think you have a point. I would rather not respond to you right now, but instead go on to my last slide, which touches on this. No, which just touches on this point. So it's very nice that you made it now. And so what I want to do now is to give a prototype of the kind of explanation that most of us working in developmental biology accept as perhaps the correct explanation up to a certain stage, of course, not the ultimate explanation by any means, for the same sort of problem, for the same sort of phenomenon that Turing looked at. And I present here a zebra stripe pattern, oddly enough coming from a fly. It turns out, interestingly, that during the development of a fly, even before it's a larva in its embryonic stage, it is like a zebra if you wish. Of course, these are not really stripes that you can see with your eye, but if you use the right kind of staining techniques, you can show that there are genes, and there's a whole baggage of them now. This is one of them. There are genes which are, now it's exactly like, if you wish, melanin deposition in a zebra's back. By the way, this is the embryo. You don't see it in outline at all, right? So it looks like an egg there. Okay, so there are genes which are not active there, active within this spatial region of a stripe, not active, active, not active, active, not active, active, and so on. And if you count there are 7 of these stripes in the fly's back. And the question is, what might cause these stripes? By the way, to give you some idea of the real fact, this could be a millimeter or 2 in size, some such thing. Okay, the currently popular explanation is the following. And I'm cheating here a little bit because this explanation applies to a different kind of stripe, but it doesn't matter. And I'm not going to take you through this slide, which is complicated enough, but I'll tell you basically what the nature of the explanation is. So it says, let us number these stripes. Fair enough, 1, 2, 3, 4, 5, 6, 7. Then this explanation says, stripes number 2 and 5 are formed where they are formed, right? Let me repeat, these are stripes, if you wish, of activity of the same gene. Please remember, so what we are looking at is whether a given gene is, in popular language, on or off. If it's on, we say that's part of the stripe. If it's off, we say that's the blank region, not part of the stripe. Okay. So the current explanation goes something like this, and I'm not literally following the screen there, but that's the spirit of the thing. It says, where you see stripes number 2 and 5, I have 2 other genes, A and B, which I know are positive regulators of the gene of interest to me. I'll call that X. So both A and B feed in positively on X. There's a third gene, C, which feeds in negatively on X. C happens to be inactive in these places, right? So both in the location of stripe number 2 and stripe number 5, positive signal from A, positive signal from B, negative signal from C. Excellent. So X goes on. So I have stripe number 2 and stripe number 5. Then the explanation says there are 3 other genes, E, F, and G, which by similar combinatorial tricks give you stripe number 1, 3, and 4. Okay? And then it says, aha, you've forgotten 6 and 7. For 6 and 7, you need this stripe itself plus something else. And there's a person by the name of Akam, who has very, very aptly said, this is an extraordinarily inelegant way of making stripes. And for heaven's sake, you could have come up with something better. But this is what the fly does. And the entire, of course, what is really elegant about this story is the experiments that were designed to uncover these sorts of truths. They are really intellectual tours de force. But that's the way the insect makes the stripe. So you see, there are 2 kinds of explanations. The kind of explanation that biologists say now, and of course, one would like to know what makes 2 and 4 active there, what makes this, that, E, F, G active here, there, and so on. But an ad hoc method of generating pattern. Superficially, this pleases the biologist enormously, because for one thing, it puts the mathematician in his place. No simple pattern. And secondly, it confirms to some kind of intuitive idea that you have complexity a la messiness, if you wish, in biology. And this is something that biologists, if not revel in, like they say, yes, this is our sort of subject, all right. Now, what I want to put across to you, and here I finally end, is the following. Maybe, and I just leave this as a question, obviously I don't know, maybe one is looking at this in the wrong way, in the following sense. It's fairly clear that genes are products of evolution. By the way, there's no one who questions evolution at all, so I'm not in the least, I hope I'm not in the least trying to suggest that. Genes are products of evolution. So there's some very complicated hierarchy of, if you wish, gene states which have led in the course of evolution to this particular kind of design of stripes, of this particular stripe. And this hierarchy has arisen, it has had historical antecedents. Clearly this hierarchy was not there in the past. There may have been a simpler hierarchy, there may have been other molecules which were doing the trick and so forth. One thing that this hierarchy of genes assures, and this seems to be something that nature, if you wish, sets great store by, is reproducibility, accuracy, predictability. One fly is like another fly is like a third fly. All of these are extraordinarily tightly regulated patterns. Errors are very poorly tolerated. This is true in any development, human development as well. Errors, especially during embryogenesis, are discarded right away and so forth. So very, very tightly regulated. It's quality control that's going on here. So the thought I want to leave you with is perhaps one thing that genes have evolved to be good at, and they do that now of course, is to be as quality controllers. And perhaps in the distant past, now it's very, very hard to say anything about this other than just wave hands. Perhaps in the distant past, you actually had, okay, some kind of embryonic structure developing, making these stripes, or stripes such as these, let's say, by a Turing kind of mechanism, with all sorts of problems, however. One of the problems being lack of size invariance, the other problem being lack of structural stability. You could change a few parameters and the whole thing would change and so forth. So it may well be that this sort of physicists or chemists model of patterning via Turing instability may really be telling you that there are things, real chemicals in bodies which have these properties. That, by the way, we know. Autocatalysis, this activator kind of property and inhibitor kind of property exists. And they probably generated the sorts of prototype patterns which served creatures long, long ago, when? In evolution. And what happened in successive generations was the fine-tuning of these patterns by genes coming into play and making sure that variations about whatever the preferred mean was were decreased less and less and less. So this is a thought I'd like to leave you with. Might be interesting. I'll just make one comment before ending. It's interesting, it's curious that one bunch of people who have taken sudden interest in Turing patterns, by sudden I mean over the past 5 years probably or 10 years, are paleontologists. So there's a great amount of activity now among paleontologists, among people who study Cambrian fossils, even pre-Cambrian fossils and so forth, to try and interpret not only the sorts of patterns you get, quite often these are shell patterns, so patterns of deposits which have later fossilized and so on, patterns that you get, but also variations about these patterns as Turing sorts of exercises. And of course the aim there is to hit or is to direct your attention not so much on the pattern itself, but on how much it varies. And then to say, look, a genetic mechanism couldn't probably have given rise to so much variation, but slight increases in size. Someone came and bit off a piece of your body, so you have to regulate to that smaller size and so forth. That could explain it. I'll stop here and take any questions that there might be.